Hey.
Where is everybody?
Food.
Are we having a talk at what?
Yeah.
Like in 4 minutes?
Yeah.
Hey. Cool.
Hey.
Hello. There you are. You're out of nowhere. Can you hear us? I think it sounds. I can hear you. There we go.
Can you hear me?
Yeah. Yeah. Cameron?
Fantastic. It looks like a great group y'all have there.
Yeah. Nice little gang here. All right. We're gonna set up Dubois. Yeah, we can just hit the screen share there. I need to. Yeah, we need to get that screen here. Should be there, right? Yeah. And then we just have to make it full screen. Can you see that, Cameron? Cameron, can you see that? You might have to exit that just for a minute. Frozen?
I don't know if y'all couldn't hear me then, but I said it so y'all were broadcasting, so I think it muted.
Oh, I see. Okay. So we'll put up the slides on full screen then? All right.
Yeah. So y'all are fine. I just if y'all don't hear from me, everything should be good.
All right. Okay.
So Prantik, will you be able to present to introduce Dubois or?
Sure.
Yeah. Cool.
All right.
Yeah, make it a little bit more cohesive.
Right. We'll start in like at 2, I guess, in a few minutes.
Yeah, definitely. So people should be joining. I'm going to set up things on this side, but I'm going to set y'all as presenters so y'all won't be able to hear me, okay?
Okay.
And just to clarify, I will not be on camera, so I can stand in front of the room.
You can stand. Do you want to introduce the person, Brian, or do you want to just read your voice over the slides? Yeah, we can show that. Let's turn off the slideshow for a bit, show the group, and then we can switch to the slideshow.
The only thing I'd ask, it's a bit long, so I'll give you a couple minutes to just read. All right. I said it by email. I don't usually walk around.
Okay, I think we should be good to go. And then let's face one for you to sound like I'm still working for seats right now. Yes. Okay, so I guess all right, so are we up? We're live? I'm not sure yet, actually. This is live.
You are presenting to everyone.
All right. No pressure.
You say 800?
800 million? Almost says American. Okay. Yeah, you're live. Okay. Okay. Hello, fellow brain hackers. We are Brainhack-NYC at Mount Sinai in New York. We have researchers here from NKI, NYU, Columbia, of course, and local folks. I'd like to present Dr. Dubois Bowman. He is the chairman of the Department of Biostats at Columbia University and the Mailman School of Public Health. He leads a research program focused on the development and applications of statistical methods for complex neuroimaging data. His research aims to improve mental health disorders such as major depression and schizophrenia, as well as neurological disorders such as Parkinson's disease, by characterizing the patterns of neurological disruption associated with these conditions. The research encompasses multiple imaging modalities, including fMRI, DTI, and PET. His research has attracted a lot of extramural funding, very successful there. And he is currently the PI of an NIH-funded research project, analytic methods for determining multimodal biomarkers for Parkinson's disease. He's also previously founded and served as director of the Center of Biomedical Imaging Statistics at the Rollins School of Public Health at Emory University. He's also an elected fellow of the American Statistical Association and serves as the intermediate past president of the Eastern North American region of the International Biometrics Society. He served as the associate editor of Biometrics and as the associate editor of the Journal of American Statistical Association. So with that, I'd like to hand the space, the ether, over to.
Thank you very much for the long and gracious introduction. I feel like I'm running for political office now. So, okay, can we go to the?
It might be, like, you want to sit at the end there? No, you're going to be screen sharing. Yeah, so you can share the screen.
Yeah.
Okay.
Terrific. So, greetings from New York to all the participants in Ann Arbor or Berkeley or Los Angeles, Miami or Mexico. I think the other sites are not going today, but I'm planning to talk to you about some of my work that establishes predictive models for Parkinson's disease. And in this particular case, we had access to multimodal imaging. So we'll kind of just take you through our work. There are a long list of collaborators who contributed in some way to this. In particular, for what I'm going to talk about today, I'd like to acknowledge Daniel Drake and Dan Huddleston. So really the drive behind the work that I talk about comes from an initiative launched by the National Institutes of Health here in the United States, in particular the National Institute for Neurological Diseases and Stroke, or Disorders in Stroke. They launched a program called Parkinson's Disease Biomarker Program, which really the name of the program conveys the major objective. And there are lots of different approaches that are being taken to this. Imaging is certainly not the sole driver or even the main driver. There are people who are looking at cerebral spinal fluid markers and things drawn from plasma. My work took the view of multimodal imaging, and so we'll present some of these ideas today. So in thinking about a biomarker, just to clarify, my use of the term biomarker will be very, very general. So the definition shown here, a characteristic that is objectively measured and evaluated as an indicator of normal biological processes, pathogenic processes, or pharmacologic responses to a therapeutic intervention. So some might view biomarkers at only a molecular level, the way I've defined it here, or the NIH working group defined it here. It can be much more general, even linking to something like behavior. And to date, there are no proven biomarkers for Parkinson's disease. So just to say a little bit more about Parkinson's, most of you are familiar, may even know people who've been affected with it, but it does impact about 1 million patients in the United States, roughly 7 to 10 million worldwide. There are a set of hallmark motor symptoms that occur: tremor, bradykinesia or slowness of movement, rigidity, gait impairments. And then there are a host of non-motor functions as well, or non-motor symptoms, disruptions to cognition, sleep, sometimes disruptions to smell that occur. There's no diagnostic test to validate PD, so sometimes you have in fact, most often you'll have the onset of symptoms and then a lag that occurs to diagnosis, where there's a process of ruling out other potential diagnoses. Usually it's a disease of the elderly, affecting people over the age of 50, but there are some instances, including of some celebrities, of early onset. And then there are some drugs, but the drugs really just try to mitigate the symptoms. They don't halt progression of the disease. So this diagram, I think, gives the motivation of the program overall, and then of our work. We won't accomplish all of this in today's talk, but just to give some framing around what we're hoping to do and the rationale. Is there a? So on the y-axis here, we see neuron function, And then on the x-axis is time. And so this vertical bar here represents the onset of symptoms, and then the second vertical bar represents diagnosis. So we see this period of decline in neuronal function prior to the onset of symptoms, and then ultimately to a diagnosis. And so from a clinical perspective, we're really then left to this space right here. So this premotor period really defines an optimal window to try to identify other markers other than the typical motor symptoms that would only manifest at that point. And what we're going to do, again, according to the title and some of my introductory words, we're going to try to stitch together information from different imaging modalities, capturing characteristics of brain structure and function, and see if we can tease out things that help to dissociate PD patients from healthy controls. So our approach here, what I'll talk about in this talk, comes from this exploratory kind of discovery phase analysis. We do, and some separate work that I won't comment on today, we are targeting some other hypothesis-driven markers, even from other MR-based imaging modalities that are not discussed. So the data that I'll talk about, we have 47 subjects that I'll talk about, the 29 PD patients and the 18 controls. I won't include the Alzheimer's disease patients. And so again, the multiple modality imaging data, as well as the demographic measures. So the first question that comes, after we collect the data, we have to decide how we're going to represent the data at a point where we're able to introduce it into the predictive model. And so this just shows a separate, and in fact, a paper that I collaborated on with some people at Penn State University on different subjects, and it shows differences in cortical thickness between PD patients and healthy controls. And so the red areas show areas where there was a high probability of differences in cortical thickness. And so we could think about, well, how do we capture information like that may be useful in a predictive model? And we won't use this directly, but it's sort of a heuristic for how we might think about representing the information. So the first thing that comes to mind, we collect all the data at a voxel level. Some of the information will be localized to a particular voxel or spatial unit. Other things we'll look at pairs of locations. And so the dimensionality just explodes. We have literally billions of possible features if we operate at a voxel level. So we'll coarsen things a bit, still whole brain, but we'll use a standard AAL parcellation, which was published maybe 2002 in Neuroimage or so, for some of the measures, in particular the white matter-based measures, so DTI. And then we wanted to refine this a bit for the functional data. And so what we ended up doing is, in a hierarchical fashion, we took the AAL 90, subdivided some of the regions, particularly the larger ones, resulting in 290 smaller regions. So these will be the basis for kind of our spatial representations of the different imaging modalities. Okay, so we target really the usual suspects. From the diffusion tensor imaging data, we calculate FA, again originally at a voxel level, and then we summarize that across the 90 regions. We also perform tractography and look at ultimately the probability of connection between different pairs of regions. The functional MR data, we have localized measures at a voxel level where we look at FALFF, or fractional amplitude of low frequency fluctuations. From a voxel level, within the 290 spatial representation, we'll summarize those measures and then compute resting state functional connectivity between all pairs of the 290 locations. So these are not the actual functional connections, but the 290 locations between which we will summarize the functional connectivity. And then lastly, we perform voxel-based morphometry on the T1 data to give estimates of density. We restrict our attention to the gray matter here, and then ultimately from the voxel level, we'll summarize those at a region level. Okay. So in summary, we have the neuroimaging features represented now at a region level that will produce these regional summaries of FA, VBM, FA-LFF. And then for the pairwise measures, we compute functional connectivity based on the 290 parcellation, node parcellation, and then the structural connectivity based on the 90 node parcellation. The reason that we use 90 for structural connectivity is simply that if you do it in a more refined fashion at 290, many of the regions don't have any white matter. So it makes sense there to use the original AAO, yet coarser parcellation. Okay, so this summarizes essentially the feature count that we have based on the parcellation scheme that I just described. So here are the modalities, the specific features that are targeted, the spatial scale based on the parcellation, and then the resulting number of features. Okay. So overall, we have over 46,000 features that stand to contribute somehow to dissociating Parkinson's disease patients from healthy controls. We're not going to stick 46,000 things into a statistical model, right? So the first thing we do is a heavy dose of screening to just try to tease out things that are worth pursuing in terms of our search for these biomarkers. And so what we do essentially is a univariate independent screening process where we look at each one of the features and its association with Parkinson's disease, and we try to, just for each modality threshold just to get sort of the most competitive players. We do something additional, and this will be kind of a recurrent theme throughout my talk today. Ultimately, what we want is to produce a set of markers that have promise for exploring further. 46,000 is a large number of features. If we were to collect data, a different set of data, and let's say go through the same process and extract 271 things, would we select the same 271? Perhaps not, right? And so what we implemented in ours is a bootstrap procedure where we actually took different bootstrap samples and repeated the screening in each one of the bootstrap samples, and ultimately retained things that fell above the threshold in at least 75% of the bootstrap samples. Okay, so that's encouraging this idea of reproducibility. So from the original feature count, we have 24 VBM that survived, 6 of the localized fMRI measures, 225 FC, 6 of the fractional anisotropy, the localized DTI-based measures, and then 10 structural connectivity pairs from the tractography. Okay, the next step is we apply a penalized regression, logistic regression in this particular instance model. And this constitutes a penalty term. So we kind of have a blend of an L1 and L2 penalty. And this parameter alpha, which is going to vary between 0 and 1, will determine how each one of those penalties contributes to the overall. In one case corresponding to ridge regression, in the other case corresponding to the lasso. This is a well-known, a well-established procedure. It's referred to as elastic net, so that it's not a development of ours and widely used in practice. So this allows us to pool the predictive strength across multiple modalities, perform shrinkage or variable selection. And so if in some cases, as the parameters are shrunk towards zero, or maybe all the way to zero, then it may tease out the important subset of predictors. It's well suited for high dimensional data. The reason we have to do this rather than a regular logistic is because we still have 270 predictors that we're including in the model, and fewer subjects to support a fit. And then this last bullet is the one that I commented earlier on. We started in the case of screening, but we'll do other things to try to encourage, again, reproducibility and robustness as constant themes throughout. All right, so this is the beginning of the story for how do we do. If we just take ElasticNet, apply it using the 271 features, how does it perform? So one other thing that we did, we used k-fold cross-validation. So you separate the training and test data, you build the model on the training data, identify the parameter estimates, and then you use the test data to see how it performs. How one constructs the training and test samples may impact your accuracy. It may also impact the variables that you choose, right? So there's some variability. Typically, people only do it once, but there's some variability in there that we wanted to at least get assess. And so we did it actually 200 times, where we reallocated the training and testing sets in each time. And so that's why you see sort of the multiple lines in this RLC curve. So sensitivity is on the y-axis, 1 minus specificity is on the x-axis. So the ideal location is in the upper left corner. So we see very, very high accuracy in general, just with a straightforward application of ElasticNet. Okay? The black trace is sort of the average of all of the 200 runs that we did. All right, so we get great accuracy. End of story. Possibly, depends on what your objectives are, right? And so for us, we weren't satisfied with that, because how do we begin to tell the story with 271 variables? So it's high accuracy, but with a hope of eventually discovering something about neurobiological meaning. We wanted to achieve a bit more parsimony than what is achieved. Essentially, there's no parsimony achieved if you just run it. You end up at an operating point here, and I'll explain this graphic in just a moment. Okay. So the punchline there is high accuracy, but you utilize all 271 variables. So what we ended up doing next was deciding to really restrict the search space for the parameters. And so in the equation that I showed earlier, the ElasticNet equation, you saw there was a parameter alpha, which is the x-axis here, and then lambda was the other parameter. And so for a grid of values for alpha and lambda, we actually repeated the procedure and reported color based on the average measure of accuracy, the area under the curve. So this black trace here would correspond to a single point, and it happens to correspond to point A in the lower left-hand corner. So you see alpha is all the way to 0, hence it's ridge regression, and all of the variables end up contributing. So it's why we get high accuracy, but no variable selection achieved. So the first thing we did was establish this white trace, and everything to the white of the right trace will have 75 or fewer variables. And then we identified this black trace. Everything to the left of the black trace will have area under the curve of at least 0.9. And this picture will look different. And in fact, I think it's useful to show things like this, because here it's a pretty smooth change, but in some cases I worry about the robustness of selecting a tuning parameter operating point right on a cliff, if you will, right? And maybe with a slight change, you end up getting substantially different accuracy. So we're going to essentially restrict our attention to that search space. Okay. So if you can memorize the locations here, this is B, C, D, A I pointed out earlier, and then E, it's just a point sort of randomly selected in the middle. These letters on these figures correspond to those points. So they're just representing different points in that space. These are what's plotted here on the y-axis. All of the axes are the same across the figures. So on the y-axis, we have the mean absolute value of the coefficients, representing all 271. And then on the x-axis, the percentage of time over the 200 different training and testing configurations, the percentage of time that the variables were kept. And so across that search space, you're going to get very, very different patterns of this scatter plot. And you see a horizontal reference line in each one. What that does is identifies the top 10% of variables with the strongest absolute value or magnitude of the coefficient. So the strongest predictors. And on the right, you can see different colors. We've color coded them. So you see mostly green. Those correspond to functional connectivity. But there are some voxel-based morphometry markers, structural connectivity. Just below the black line, the horizontal line on the panel to the left or to the right, you see another structural connectivity in VBM. So that's an arbitrary placement. Had we put it a little bit lower, we would have included those. But remember, we're doing this multiple times or across multiple points. And so for other points on the grid, some of these points may fall above, and ones that are above now may fall below. Okay. So what we found was very high consistency across all of the different points on the grid for variables that fell above in the top 10%. Okay. And so the second column, so the first column in this table just gives the name of the feature. So the FC just represents functional connectivity, the SC structural connectivity, and tells the corresponding regions. And then we have two just localized VBM measures. The next column represents the proportion of time, the proportion of locations on that grid that it was in the top 10%. So for many of them, across that entire search space, you don't get any change. Like the strong players remain the strong players. And even for the ones that there were 16 of 23 here that were consistent 100% of the time. Okay, and the others were shown are above 90%. The x-axis measure were the percentage of time that they were kept across at a particular point on the grid. Sounds like someone got swallowed. A bit scary. So maybe I'll stand on this side. So these range quite a bit in terms of the selection rate. With some being as high as 100, others, I think the lowest actually. This must have only been kept in a few times at a particular point. Okay. So again, what you're hearing is an application of elastic net, but some variations that again really just try to target robustness to the tuning parameters. No matter which one of the locations that we choose in that area, we get a sense of how the different variables emerge, and then the reproducibility, and all of this was to achieve some parsimony. And on the right, we have the direction. So when you shrink the parameters, they're not necessarily comparable in each case because there's some bias introduced, but you do get an indication of the direction. And so we can tell for the structural connectivity, it's reduced structural connectivity between these two regions in PD patients relative to the healthy controls. All right, so this is where things get difficult to move beyond. So we have a set of 23 variables that seem like they're very significant contributors to the predictive model. Can I tell you, the exact 6 or the exact 7. It's actually hard to do that. There are subsets of as few as 3 variables that give perfect separation between the groups. And so these are the 6 models. And we see the amygdala show up in a few of these. The thalamus shows up in FC, a couple of the FC measures. So we get an idea of the 23, but for the groups, the groups I think are fairly well separated. We're talking about PD patients, although early to mild to moderate, they still have PD and healthy controls, and so it's hard to really tease out anything additional in terms of which one of the variables or which subset of the variables are the stronger contributors among the 23. But this is an example of just 6 even different model combinations of 3 that yield perfect separation. Okay, so here's what I think we've learned. So this is, again, I view this as like a discovery phase, where in exploratory analysis, we're just trying to identify if there are some drivers that help to dissociate the PD patients from healthy controls, and we hone in on 3 of them. That represents a really, really small proportion of the original features, with even smaller subsets achieving perfect separation. These now need to be examined further, right? So we're going to have to collect different data sets, and we're in the planning stages of doing this. But to take those 23, see if they emerge in a different data set, see if we can use them, let's say, for other purposes of monitoring things like progression. So for people who have Parkinson's disease and you follow them over the year, people who progress and who don't progress, those groups may not be as clearly separable as PD patients in healthy control. So to do more pressing, ask more pressing questions that might help to even tease out among the 23, are there even a smaller subset? Where the field is right now in Parkinson's disease, there aren't any neuroprotective treatments that are sitting on the shelf that can be used for that early intervention. So, I hope that one benefit of my work longer term, the work of others in the Parkinson's disease biomarker group, is that they'll help to even inform future clinical trials, so we can develop the neuroprotective treatment. So we're still at a really, really early phase. And then the last thing I'll say is, although the work that I talk about is specific to Parkinson's disease, and that's what drew my interest, the general ideas are extendable to other applications. And I just put a few here, maybe predicting treatment response and depressed patients who are treated with different antidepressant therapies, determining the risk of progression from mild cognitive impairment or MCI to Alzheimer's disease, or dissociating PD from related Parkinsonism disorder. So that lag between the onset of symptoms and a diagnosis, in part, is to rule out other sort of related diseases or disorders that share some of the same symptoms. And so if we could use these markers or other things, or the methodology even, to try to identify things that help to separate those groups more reliably, that would also be beneficial. Okay. So that's the end of my talk. I'd be happy to take some questions.
Okay. I'm not sure if we can actually take questions.
Okay.
Other sides. Cameron? I have a question, general. So what do you think, do you think a multivariate biomarker is clinically translatable?
Translatable in terms of implementation, or translatable in terms of
implementation in a clinical context, where if can you imagine a future where, based on your model selection, you say, acquire, these 4 scans and look at these n areas, then like add them up and like take a score. And this is like, do you think, that's what?
I do. I mean, so the end point of my talk doesn't put us there, right? But if we can project the development and thinking out, and arrive at a place where we have, a small subset of measures that are consistent, that we do think maps on to some of the pathophysiology of the disease, maybe some indication that they arrive early in the process before the onset of motor symptoms. I do think there's promise there. That I'll make reference to a non-imaging aspect of the same research that I'm doing, where we're actually using things like electronic medical records. And one of the things we find, and that work's ongoing, we're not finished with that either, but one of the things we find is that as early as like 2 years prior to a diagnosis, that some patients are diagnosed with having sort of impaired or involuntary movements. And I mentioned that just to think of almost like a coupling, right? So maybe it's not that every person who walks into a clinic is recommended to have multimodal images done, but from a cost perspective, that wouldn't be feasible. But if maybe in combination with some other indicators, some people are viewed to be at high risk, then they could possibly be referred. So from that perspective, I do think it's possibly trans.
I mean, I guess like the model selection and the, in terms of finding a more parsimonious explanation of the classification, it implies, I guess, that kind of trick, right? That we handle things. In terms of like translatability.
Yeah. Yes.
Do these areas get worse as, do they like become more noticeable as the disease progresses?
I don't know the answer to that, because these data that we have don't let us address that. There's a model out there that was developed by Brock and Brock that suggests that there is phased pathology, that there's certain structures like the locus ceruleus and substantia nigra that are impacted first, and there's sort of a phasing process. But the extent to which, based on the data that I have available, the things that we identify, how they get worse over time, we don't know. We do have access, or we will have access actually to data where we can actually evaluate progression. So scans are taken out 1 year apart, and we'll be able to answer that question, but we can't answer them based on the data that we have here.
So this data is all from like around, at what time point of the interview?
It's the inclusion criteria just specified that they were early to mild to moderate, right? And so it's whatever the snapshot is for that particular patient in that process, but we don't follow and rescan for these data.
There's a question from Berkeley. I have a question. Do you have a data set for people who are genetically predisposed for Parkinson's, but who don't express a disease later in life, and might be interesting to run a similar classification between or within Parkinson non-expressor and those who do express Parkinson's?
Sounds like a terrific study. If you have those data, please call me. There is a lot of genetic work, and it's not even clear, like as with many diseases, I think this is true in movement disorders, and we have some specialists here who can maybe speak as well on this, but that not all, everything that we call Parkinson's disease may not have the same cause or drivers, and that there may be some, particularly like the early onset, that it may be more likely that those are genetically driven than some of the other forms of Parkinson's disease that occur. So that sounds like it's a fantastic question. We do not have data to answer your question explicitly. We do not have data like those to try to tease those out, but it would be, I think, a really, really important study to do.
Okay, cool. Thanks, guys.
All right, thank you.
Thank you much. Okay, Cameron, I think we're done here, if you can hear me.
Can you hear me?
Yep.
Okay, fantastic. Yeah, thank you very much. That was a fantastic talk. I'm going to stop this broadcast now, and then we'll connect with Eleftherios.
Great.
See y'all again in a minute.
Right.